Hello everybody, this is Mr. Dwight Doodles, and this time around we are looking at B3 infection and response and its defense against disease and vaccination. So let's dive straight in. Let's dive straight in with the diagram that I always draw, and it's the thing we should always see when it comes to biology. We have our blood vessel here and we have our cell. The cell needs to function, and that means it needs to respire aerobically to release energy, move, maintain the warmth for enzymes to work, and build larger molecules. Now, let me introduce you to the villain of this story. This here is a bacterial cell. A bacterial cell is a prokaryote. They don't have nasty faces like this, but what they do have is this unique three-dimensional shape, and this is called an antigen, and this word you do need to know. Antigen. It's a protein, and it sits in the cell wall of the bacteria. It's unique to that bacteria. So once the bacteria has got into your bloodstream, it is now inside your body. There it is. It's not yet causing havoc, but what it now does is it releases a toxin. And these toxins can move into your cells, or they can attach to your cells and proteins within your cell membranes, and they alter the functioning of your cell. And this is where you'll experience symptoms, which could be a runny nose, poorly throat, or a severe as shutting down your respiratory system. So now you've got an issue. And the other thing bacteria do extremely well is they replicate. And they replicate not by mitosis, because that would involve chromosomes, which bacteria do not have. This is called binary fission. And so now we've got three of the little tearaways, and they're all releasing toxins, and you're bedbound with lots of tissue because you've got an extremely runny nose. So what happens next? Well, let me introduce you to the second character of our story. This here, there's not to scale. This here is a lymphocyte. Now, I've drawn some glasses onto my lymphocyte, and obviously they don't have glasses, but this lymphocyte, there is a meaning to the glasses because it allows it to see. And what it recognizes is the bacterial antigen. And this triggers a process. So let's put our lymphocyte into our blood vessel. Now, the occurrence of the lymphocyte recognizing the antigen does take some time. So this could be up to 3 days. And in that time, the bacteria cells are divided by binary fission, loads of toxins, you're feeling really poorly. But once this time frame is up, the lymphocyte says, again, it doesn't have a mouth and doesn't talk, but what it does is it releases something called an antitoxin. So let's do these little, and the antitoxin masks the toxin. It destroys the toxin. So this then begins to take away the symptoms that you may be experiencing, but it still leaves the bacteria there. So number one, we've got release of antitoxin. Prevents the feeling of the toxin and relieves the symptoms. The next thing it does, the most important thing it does, it releases another protein, and these are called antibodies. Now, these antibodies attach to the antigens of the bacteria. Just going to move it down slightly here. And so what it does is it clumps all of the bacteria in one said location. So there's our 4 bacterial cells. The antitoxin has masked the toxin. And now what happens is the antibody has attached to these little tearaways. It's clumped them all together. It's prevented them from dividing by binary fission. And then more importantly, it sends out a signal to another type of white blood cell. Now, this white blood cell is the destroyer. This is called a phagocyte. And what the phagocyte does is, once called upon by the antibody, comes along and it ingests all of the bacteria and the antibody. Enzymes within the phagocyte that undergoes phagocytosis destroys said bacterial cells, and it then places the antibody into its cell membrane alongside the lymphocytes. They now have a memory, and these are known as memory cells. That is the process of what we call active immunity. So let's dive straight into an extended response 6 marker and what we would expect to see in this question. So it's a describe. If pathogens do enter the body, into the immune system, the immune system tries to destroy the pathogens. Describe how the immune system defends against disease. So I highlight the word describe. We all now know that it is the list the events in a chronological order. So let's just do that. Firstly, what is the name of the cell that recognizes the bacterial infection? This is a white blood cell. So white blood cells destroy infection. But what is infection? Well, infection is bacteria, viruses, and fungi. Okay, so how do they do that? What do they do? Well, firstly, lymphocytes recognize antigens on bacterial cells. Lymphocytes release antitoxins that mask the toxin. Lymphocytes produce antibodies. What do these antibodies do? Antibodies attach to antigens. Okay, so what effect does that have? Well, it stops binary fission. It clumps bacteria together. What else does it do? Well, it then stimulates phagocytes to undergo phagocytosis. This is where they engulf the bacteria and antibody and create memory cells. That's a nice describe. Now, if we look at another type of immune question, we get given a giraffe and our command term has changed to explain. So we know that explain is state because. Let's highlight the 2 key terms that it is telling us to talk about. Explain the differences between antibody production after the vaccine injection and after exposure to the measles virus. Explain the differences between antibody production after the vaccine injection, which is here, and after exposure to the measles virus. So here we go. Concentration of measles. Explain the differences. First, you've got to state the differences. So exposure to the measles virus, there is a higher concentration of antibodies produced. Apologies, I didn't recognize there. I didn't read on. It says data. You've got to use data. So you would have to quantify a statement based to substantiate that. So if you draw your line here across 7.2, and what does it begin at? That's 0.8. So you'd work out what the value is between those two. So you're quantifying the statement, or you could say it is X amount times higher. I believe it's 9. If we look, that one takes that time in order to produce those antibodies, but it's much quicker on this time. So number 1, there are more antibodies produced. Quantitative statement, so from 0.8 to 7.2, that's our value. Antibodies are produced sooner. Also, if you look, they decrease quite rapidly, but on here they stay high for much longer. So the antibodies stay at a higher concentration longer. So there's our statements. There's our statements regarding the graph, but it does say explain. So now we need to say because. Well, white blood cells, if we refer to our previous question, what do? Well, they recognize, so white blood cells, lymphocytes, recognize the antigen of the measles virus. And what do they do with that? Well, here we go through what occurs next is that they release antibodies, which is stated up here in the giraffe. And those antibodies are specific to the virus. And the key there is stating by using the graph and numbers that after the measles exposure, when you've had the vaccine, it is faster, they stay higher longer, and the production is rapid. Those three things with a vaccine is key.